Hi, I'm going to show you how to take some publicly available data from Geo or some other repository and basically get it ready for a lab meeting in about 20 minutes. And what we're going to do here is a very basic kind of vanilla walkthrough. We're not going to fiddle too much with the dials or any of the settings, and I'll touch on essentially what we're doing. And at the end of this, the idea is that you'll have created some clustered QC'd single cell data that you have annotated and classified somewhat. Now the script is going to be available and I'll include it at the bottom of the video. Now I don't profess to be an expert. I'm an enthusiastic student of single cell RNA-Seq, but I'm not the one writing a lot of these packages and functions. But I'll show you how to cobble them together to get results quickly. So the data we're going to be looking at is from a paper that I actually published recently because I'm somewhat familiar with it, so I know what to expect. There's a link here to the Geo page. Now when you do download something like this from Geo, what you'll find is 3 files. Generally speaking, you'll have a matrix, in this case a sparse matrix, which is more useful because it's a more efficient way of storing it. You'll have your genes or your features, which are the transcripts that you'll have sequenced, and they essentially make up the rows of this matrix, and the barcodes, which are the cell barcodes, which make up the columns. Now, for the built-in function that we're going to be using to work, these need to be renamed ever so slightly. So they're renamed as follows. You basically need to have barcodes.tsv, features.tsv, and the matrix, rather than the full title, which includes the Geo code. Now obviously when something comes off the sequencer, it's in a fastq file, and there's a whole alignment step that needs to be done, but I can do that in a different video if you want, but that's actually a lot more involved and a lot more complicated, and often when you're using other people's data, you're not going to need to do that. You can generally pull from a repository a matrix of cell gene counts. So I'm going to assume a basic level of familiarity with youre, but I'll talk you through some of the trickier bits. So to begin with, you need to ensure you have the correct packages open. So start a new folder and make sure you have installed the packages you see listed here. Specifically the first 3 are the most important, but you'll likely need all of these if you want to follow everything through just as I provided the code. I just explained what each package does here, and I'm going to use a surat package to take us through the workflow. There are other ways to do this. You can do it SC3 for example, or you can do this in Python, which is excellent. The main reason I'm using surat is simply familiarity. It's how I learned and it's what I'm most familiar with. There are advantages and disadvantages to each, and indeed to using youre itself rather than working in Python. The main disadvantage to youre is that it loads its objects directly into the RAM. So if you were to have a data set that is 15 gigabytes and you've got 16 gigabytes of RAM, you're going to run into major problems. So you would need to then do that on a compute cluster, for example, or on a bigger machine with more hardware. So you downloaded your data from Geo. Most of the time it's going to come as a tar.gz, so you'll need to unzip that twice. With 7-zip is a good free program that does a good job of doing that. And then make sure the file names are as I've just shown you, and that is barcodes.tsv, features.tsv, and the matrix.mtx. You'll need to have the matrix package installed and loaded to open that. And then you essentially use a very, very convenient function from surat itself, which allows you to read that pre-formatted 10x data. And I'm simply going to read it in. You can see it's a large sparse matrix, and it's actually quite a small file size. There's only about 5,000 cells here, so things should move quite quickly, and I'll accelerate any things that involve computation. Then the next thing is that if you have a look at this, you'll see that the rows, or the features in this case, are ensemble IDs. Gene symbols are obviously far more readable for a biologist, and I'm clinical background, but the problem with translating and mapping ensemble IDs to gene symbols is that not every ensemble ID is going to map to a gene symbol. Some will map to 2, some will map to none, and they are more unique, and it's much more correct to run your analysis fully using the ensemble IDs and then find some way later to convert to gene symbols to make them a bit more human readable, which is quite easy. If you're just doing something like a differential expression analysis, you can easily convert your matrix at the end. It's somewhat trickier when it comes to changing the labels on plots, but it's entirely doable. But just for convenience here, I'm going to show you how to change the symbols at this stage, and anything that doesn't have a clearly mapped symbol will keep it as an ensemble ID instead. Some people will suggest you use biomart. I find it somewhat cumbersome. Instead, my approach is simply to use, this would be HS in the case of humans, the murine databases. You can select it using key pairs. In this case, my key is the ensemble IDs, and I'm going to output a symbol of columns. So run that, and you can see that some match beautifully, others don't. So what I'm going to do is, when they have a symbol generated, I'm going to take it, otherwise I'm going to default back to the ensemble using an if-else statement here. I'm going to put that into a column called final. And then using this very simple loop, I'm essentially going to replace the row names within that matrix with our new column final. And then to create the final Surat object that you're going to be working with, I'm going to run the create Surat function that comes as part of that Surat package. The object that it's acting on is that.sham, and reviewing the object you see that you've created an object of class Surat, and those are the correct features and samples. So your best friend is the cheat sheet here, which is a number of the essential commands that you're going to keep returning to, and that I often have difficulty just remembering off the top of my head. Now the next step is QC. Now there's going to be a number of QC steps before you reach this point at the level of alignment and so on. Okay, so at this point, a fairly typical basic quality control step would be to filter based on three features, and that is the percentage of mitochondrial transcripts that make up the overall number of transcripts in the cell, as well as the number of unique features, the number of transcripts in the cell in total. So to generate the percent of MT features, you can simply run this very convenient function here, remembering that in the case of humans this would be all uppercase. You can just see the row names here. This is what they look like currently. So to confirm the range is very low, I think I pre-filtered this previous step. The variant plot will let you see what you're dealing with, and you may decide thresholds that are appropriate then based on your own data, and each data set tends to be different. So in this case we're just going to run with Surat's default settings here, but I found in the past when I'm dealing with my raw data that I would be quite permissive in some cell types of percentage of mitochondrial genes if they're extremely metabolically active, like proximal tubular cells, for example. So we're going to run that, and at this point we're then going to normalize the data. Now there's different ways to normalize the data. If this were a plate-based protocol like SmartSeq2, you may have ERCC spike-ins, which you could use to normalize to. In this case, and the role of normalization, of course, is to counteract a lot of the technical noise and bias that's introduced into each workflow. And the default Surat normalization is log normalize, which basically normalizes the feature expression measurements for each cell by the total expression, multiplies it by a scaling factor, which I think is 10,000, and then log transforms it. Having read lots of recent publications, most people seem to be very happy with this approach as well. You tend not to see much variation on this, so we'll follow suit. The next step is to find the highly variable features, and this is a way really of trying to cut back on the background noise, focus in on the genes that have the most variance. It seems to generate cleaner results, so you amplify the differences more. But these are the genes that we're going to then subsequently use for generating PCAs from and further dimensionality reduction techniques. Default, they just grab 2,000 of those, so we'll just run in default. It's rare that you need to change that actually. Correct the order of things there, that's incorrect. We can take a sneaky look at to see what our top variable genes are here in top 10. Let's plot them out and label them. We've got a fairly recognizable bunch of highly variable genes. CD74 is HLA class 2. So you can imagine that there's going to be immune cells as well as epithelial and non-immune cells. So this is obviously going to be quite variable across the data set. Similarly, things like UMAP, which mark up particular tubular cells, and aquaporins, and these are all very, very characteristic of certain cell types. They all are, actually. So it makes sense that they'd be very high in one and down in another, hence the higher variance. The next stage in pre-processing then is going to be scaling the genes. And you could scale just the genes you're going to be looking at, but we might as well just do them all in something of this size. What you're going to end up with here is mean expression of 0 and then variance of 1, which means that those highly variable genes that we've seen here and others aren't going to completely just dominate all of the signal. So boom, boom. So next we're going to run printable component analysis on our highly variable genes first, and then store them inside in the object. We're going to use these to do further dimensionality reduction. You could do a tSNE or UMAPs using them, which tends to produce much cleaner results than doing the UMAP on all of the genes, where everything is really standard. You can look through each of your principal components and see what you can pull out the loadings if you want. There's going to be a point shortly where you'll need to decide how many dimensions you're going to take forward into subsequent dimensionality reduction, so how many principal components you'd like to use to feed into your other analyses. The vignettes that Sura provide have various ways of exploring that number. It's generally going to be between 10 and 20, and in every data set I've looked at about maybe a dozen or so single cell data sets at the moment. It seems not to make a massive difference, so experimentation is one way to do it. A second way to do it, and it's by far the fastest, is just doing a quick elbow plot, and you can see that when things start to level off is kind of generally where you draw your lines. So somewhere between 15 and 20, you can just, you can see here, you're getting diminishing returns as you make your way down in terms of the standard deviation. So we'll try that, we'll take 20. So we're now going to take these 20 principal components, and they essentially are summarizing the data. It's the high level summary, or a higher level summary of what we're dealing with in terms of gene expression, and we're going to just feed that into a clustering algorithm. So Surat's native clustering is a graph-based clustering. And the find neighbors function is going to be the first step in that, where you actually construct a k-nearest neighbors graph. I'm going to give them 20 principal components. They're going to generate that graph, and we're then going to use that to identify clusters within that space we've created. So the next step then is to find the clusters or communities that exist within this graph that we've just connected. Now, the default method here is a Louvain algorithm, and what that does is it tries to optimize the number of clusters that it identifies within that graph and tries to optimize the modularity of that. The bar stool explanation of modularity, because I'm not a mathematician, is that it is one way of measuring the structure of a graph and of the clusters within it. It's a way of measuring the strength of partitioning into those individual clusters, and the high modularity means that they're tightly knit and different from each other. Now, the resolution parameter here is a tweakable dial that you can play with, and you'll see that if I run with a resolution of 0.1, for example, it finds 8 communities within the graph, so 8 clusters in my cells, whereas if I increase it to 0.5, I have 11. I know from having looked at this earlier, because I cheated, that 0.1 seems to work reasonably well. I'm expecting about 8 cell types to be found here. This is an iterative process, so it's around this point in the workflow that you're going to start experimenting, running all the way through to the end, seeing what you end up with, and then coming back if you are missing cell types that you're looking for, and so on. And I want to do some further dimensionality reduction. I'm going to construct a UMAP. I'll have a link at the bottom to the paper and explanations of how it works, but it's a very, very fast and efficient way of further reducing the dimensionality down. You can create some really quite elegant structures from it. What we'll then do is project our clustering on top of that, and if everything has gone according to plan, what you would hope is that they roughly align with each other, so that discrete geographical islands on your graph will be discrete clusters as well. So at this point I tend to use the same dimensionality reduction as I did in constructing the KNN graph. The 3 parameters that follow are all quite tweakable, and I'll show you an example of what happens if you change the number of neighbours and the spread and the distance and so on. So we'll run it first. You can see that when I ran the number of neighbours at 4 with a spread of 5, appreciate that I was also messing around with the cluster number, and it looks pretty chaotic. Dialing those back then results in something that looks a bit more like this, which to me has a lot more face validity and certainly is a lot cleaner and tighter. I feel much more convinced that these cells are similar to each other than I do from looking at some of these. So you can review the images before you export them here within RStudio. To me that passes the eyeball test. The next step is that you're going to want to find differentially expressed genes or marker genes for each of these clusters, because ultimately you want to know what they are and then start exploring the differences between them. And again, it has a convenient function for letting you do this. Yeah, the default is the Wilcox rank sum test. You have the option of using others, so ORC is another one that can be interesting. You can use that with the test argument, for example. We'll leave it blank and use Wilcox. Other important arguments that are worth noting: only positive, so this will only give you positively expressed genes in each cluster. I actually think it's quite useful to know what isn't expressed often, so I always leave that as false. And then the minimum percentage is the minimum percentage of cells within that cluster that is going to express a gene for it to count in your analysis. The higher this is, it will need to be more common, more ubiquitous, but you do get quite a lot of dropout in single cell workflows, so actually, don't push it higher than 0.25, I would have thought. Going lower will help you capture rarer transcripts, but at the cost of time. And you can imagine that the log full change threshold is going to have a similar effect. So at this point I'm going to run this, I'll speed things up. It's worth noting that when you run this function, you're basically comparing your cluster to all of the other clusters. If you wanted to compare just one cluster versus another, rather than running find all markers, you would use find markers like this. Next, it makes sense to just create a second vector, which we can then use later for a heat map. Using the tidyverse, just piping it through, you can group it by cluster and just pull out the top 10 by average log fold change, like so. Heat map, which takes a while to generate, will appear. Here is a large, saved, prettier version. You can zoom in and have a look. I quite like the Viridis palette. I'll have a link below to the Viridis lecture. It's a really, really nice package, and I really like the science behind why this palette is optimal rather than something like Jet, for example. All that's left then for you to do is to assign a gene set enrichment analysis rank. I find this is far better than things like over-enrichment analysis for when you're looking at gene ontologies. There's a number of different ways to generate a rank. The way I have found, and the way that others in BioStar's forums seem to be using, is this formula here as follows. So assign a rank to each gene. It makes a lot more sense really when you're comparing one cluster to another as above, rather than one to every other cluster. But I would suggest webgestalt.org as a wonderful, really convenient way to output gene set enrichment analysis really fast. Next, you might want to look at some specific genes, because we really want to classify these and say, well, what is cluster 0? What type of cell is it? And there's lots of ways you can do this. You can do this in an automated fashion using packages like Single or Garnet, where you'll be comparing this to other data sets or an atlas if available. But sometimes it's faster, and I think a bit more accurate even, to just use your brain and take a look at what some of these clusters are expressing. So the way I like to do that is to look at the heat map, which gives you obviously what the top by fold change genes are. Also, I happen to know that, for example, these genes demarcate specific cell types within the kidney, which is what this is. We can create a feature plot here or a dot plot, and I'll show you what they look like now. So this will be a typical feature plot, SCL3184. So you can see it's quite ubiquitous, but it is proximal tubular really, whereas CX3C01 may be macrophages, mesenchymal, PDGFR beta and so on, and not a whole lot of that there, but this is actually a sham kidney, it's uninjured, so not particularly shocking. Sometimes it's a bit easier to read that off a dot plot, like so. I find it's often easier just to generate violins, dots and feature plots, which is the first one you saw, and just try and kind of triangulate what you're looking at. Combining that with the heat map should hopefully let you deduce what each one is. If there's one in particular that you're not sure about, you can always just get straight into the differentially expressed genes. Don't use Excel, it changes gene names to dates sometimes, you should probably know that. And you can start looking through the results. So in cluster 0 here, you can assess what were the genes that were most expressed, and you may begin to be able to work out what it is. And then finally, in terms of actually renaming them, the function is extremely simple. Rename idents. You can input 2 vectors or just do it manually, like I've done here, which finally will let you pump out a Dimensionality Reduction plot. In this case, I've gone for a UMAP with the cluster labels attached, like so. And honestly, the hardest part of all of this is probably classifying the cells at the end. It definitely helps to have some sort of biological insight into what's going on and familiarity with the tissue, or being friends with someone who knows that. A pen and paper is sometimes quite useful as you just start iterating through the process, trying to account for all the cells you would expect to be there. And there's definitely a degree of artistry, I suppose, when you're trying to decide how many clusters should there be, because you want to be exploring, perhaps looking for novel cell types, but equally you need to be confident that you're finding all the cell types that should be there. And if you push your resolution too high, you can generate, as you saw here, you can generate 20 cell types. But I'm almost certain that there's not 20 discrete phenotypes of cells within this data set. Yet you could easily start waving a flag around saying you've identified novel cell types based on this graph. So you need to move that slider to find that balance between identifying what you know is there and perhaps just pushing the boundary a little bit to see is there anything else there. There's lots of subtlety to that. For example, the algorithm's tendency to over-partition the large clusters. I've tried to keep that to a minimum here, but we're right to push the resolution a bit higher. No doubt this will over-partition into lots of different subtypes, which may or may not be biologically meaningful. I'll actually do that now to show you. And you can see the majority of the additional clusters has appeared into the large island of cells. Whether or not these represent biology, I would be suspicious that they perhaps don't. But again, if you can go to the bench and validate that, then actually perhaps they do. It's up to you to prove it. So that's essentially how to take some publicly available data, run it through a very basic single cell script, generate some graphs, differentially express genes, classify the cells. You could then share this at a lab meeting, or if it's your own data, this would be a useful figure 1. Obviously I've glossed over all of the math and statistics, and I haven't gone into any great depth about any of the functions really. Just wanted to get it up and running, quickly smash through, and then the real learning I found occurs when you start going back, iterating through, lifting up the hood and seeing how the various algorithms and functions tick, and what you can change, what you can push and what breaks them. This is the simplest way to run through a single cell workflow. Hope that was helpful. Bye.